Hello students, myself Dr. Prasoon Hara. I will take second paper of M.Sc final in which I will take up the bioinorganic portion of it. Let us start with the nitrogen fixation. Biological nitrogen fixation. It is an extremely important process in the soil carried out by the several group of bacteria and actinomycetes, able to absorb elemental nitrogen from the atmosphere and combine it into compounds that serve as a nutrient from the plants. Following the initial discovery of Clostridium and Azotobacter by the Vinogradsky and Bizernek, an increasing number of nitrogen-fixing organisms were isolated. At the beginning of 1970s, spirillum-like bacteria of the genus Azospirillum were found associated with many cereals and forage crops. The level of nitrogen fixation is determined by the several factors, including soil temperature. Azospirillum species thrive in more temperature and or tropical environments. The ability of the host plant to provide a rhizosphere environment low in oxygen pressure, the availability of the host photosynthetes for the bacteria. Biological nitrogen fixation is the term used for the process in which nitrogen gas from the atmosphere is incorporated into the tissue of certain plants. Only a selected group of plants is able to obtain nitrogen in this way with the help of soil microorganisms. Phosphorus and nitrogen are usually limiting nutrients because plants require large amounts of them on a daily basis. A soil nutrient is limiting supply results in stunted growth or a lower number of plants in an ecosystem. Now the next is mechanism nitrogenases. What are the nitrogenases? These are the enzymes which are mainly involved in hydrogen production during the biological nitrogen fixation. It consists of metalloproteins and can be divided into three types such as molybdenum nitrogenase, vanadium nitrogenase, vanadium nitrogenase known as VNF, and Fe nitrogenase known as ANF. Among these, molybdenum nitrogenase containing two proteins such as dinitrogenase MOFE protein and dinitrogenase reductase Fe protein, and it has been well characterized enzyme than others. It should be noted that the photosynthetic bacteria of R. palustris CGAOOG is the only organism known to possess all the three nitrogenases. Nitrogenases are highly sensitive to molecular oxygen and ammonia. These anaerobic condition and ammonium removal are essential for hydrogen production via photofermentation. The metabolic pathway of the hydrogen production by different types of nitrogenases is described below. Molybdenum nitrogenases N2 plus 8H plus 8 electron plus 16A. V nitrogenase N2 plus 12H plus 12 electron plus 24A. Fe nitrogenase N2 plus twice H plus 24 electron plus 48A. The next, you can see the diagram. This diagram showing the complete nitrogen cycle among the animals, plants and the soil or the earth. Here is the symbiotic nitrogen fixation, nitrogen volatilization, dinitrification, nitrification, runoff erosion, leaching, plant uptake. Now what is nitrification? Nitrification is the biological oxidation of ammonia to nitrite followed by the oxidation of nitrite to nitrate. The transformation of ammonia to a nitrite is usually the rate of limiting step of nitrification. It is an important step in the nitrogen cycle in the soil. Now, what is denitrification? Denitrification. It is a microbially facilitated process where intake is reduced and ultimately produces molecular nitrogen through a series of intermediate gaseous nitrogen oxide products. Now, one term is there, runoff erosion. What is runoff erosion? Yeah, runoff erosion, it is the process results in the transfer of water or soil from field to adjacent lands. These processes also transfer plant nutrients, that is nitrogen and phosphorus, and pesticides, PPPs. Pesticides runoff represents the most important diffuse source entry route of PPTs into the surface water. Now the leaching. What is leaching? It is the process of a solute becoming detached or extracted from its carrier substance by the way of a solvent leaching. Can occur usually seen from the plant substances, inorganic or organic, solute leaching in the soil and in the decomposition of the organic material. Now the next is mechanism. Biological nitrogen fixation can be represented by the following equation in which 2 moles of ammonia are produced from 1 mole of nitrogen gas at the expense of 16 mole of ADPs and a supply of electrons and protons. Here is the nitrogen plus 8H plus 8 electron plus 16 ATPs is equal to twice NH3 plus H2 plus 16 ADPs plus 16 PI or inorganic phosphorus. This reaction is performed exclusively by prokaryotes, the bacteria and the related organisms, using an enzyme called nitrogenase. This enzyme consists of two proteins, an iron protein and a molybdenum iron protein. The reaction occurs while nitrogen is bound to the nitrogenase enzyme complex. The iron protein is first reduced by the electrons donated by ferredoxine. Then the reduced Fe protein binds ATP and reduces the molybdenum iron protein. Molybdenum iron protein donates electron to the nitrogen producing diazine. In two further cycles of this process, each requiring electrons donated by the ferredoxine. HNNH, that is diazine, is reduced to hydrazine. Depending on the type of microorganisms, the reduces ferredoxine, which supplies electrons for this process, is generated by photosynthesis, respiration, or fermentation. Now the reduced ferredoxine, here is the oxidized ferredoxine. Reduced Fe protein 4 ATP, oxidized Fe protein 4 ADPs. Oxidized MOFE protein gives reduced MOFE proteins. Here is the nitrogen, here is diazine. Here NH2, hydrazine, diazine, then twice ammonia, NH2. This is a remarkable degree of functional conservation between the nitrogenase protein of all nitrogens nitrogen fixing bacteria. The Fe protein and the MOFE protein have been isolated from many of these bacteria, and nitrogen fixation can be shown to occur in cell-free system in a lab when the Fe protein of one species is mixed with the MOFE protein of another bacterium, even if the species are very distantly related. Here is the examples of some nitrogen fixing bacteria. This is free living and these are the symbiotic with plants, aerobic and anaerobic. Aerobic in presence of oxygen, Azotobacter, Biserinicta, Calocibella, Cyanobacteria, etc. And among the anaerobic bacteria, Clostridium, Desulfovibrio, purple sulfur bacteria, purple non-sulfur bacteria and green sulfur bacteria. Symbiotic with plants, legumes, rhizobium, other plants, Frankia and Azospirillum. Now the next is nitrogenase enzymes complex is highly sensitive to oxygen. It is inactivated when they are exposed to oxygen because this reacts with the iron compound of the proteins. Although this is not a problem for anaerobic bacteria, it could be a major problem for aerobic species such as cyanobacteria which generate oxygen during photosynthesis and the free living aerobic bacteria of soils such as Azotobacter and Biserinicta. These organisms have various methods to overcome the problem. For example, Azotobacter species have the highest known rate of respiratory metabolism of any organism, so they might protect the enzyme by maintaining a very low level of oxygen in their cells. Azotobacter species also produce copious amounts of extracellular polysaccharides. By maintaining a water within the polysaccharides slime layer, these bacteria can limit the diffusion rate of oxygen to the cells. And that's all. I end up with